Hello, my name is Andrew Roulard, and this is the third part of the lecture on resources for bioinformatics and machine learning. This next resource is called KINOMEscan. This dataset is being generated as part of the LINKS project, which stands for Library of Integrated Network-Based Cellular Signatures. The KINOMEscan project is quantifying in high throughput the inhibitory effect of many different drugs on kinases. There are other resources that also have this kind of data, which is providing information about the activity of drugs against proteins. The KINOMEscan data in particular can be accessed by going to this website shown in this slide. And this is a useful data set because we can search for drugs that inhibit the activity of a kinase or a group of kinases. On this next slide, we see how the KINOMEscan data is laid out when you download it. This is a bit of an unusual format. Basically, a single data point is specified in a series of 3 rows in this file. You see we have 3 different columns highlighted. The left column is telling us which drug was used in a particular experiment. The middle column is specifying which kinase was tested. And then the third column gives us information about how much inhibition of kinase activity was measured for that particular pair of drug and kinase. So we can take all this data and rearrange it into a matrix, just as we've been doing for all these other resources. And this next slide is again showing us a heat map of this network that we can now create, where we're connecting kinases to drugs that have a strong inhibitory effect on their activity. And this gives us a nice overview of how these drugs are working, whether they're broadly inhibiting many kinases or working specifically on just a few. The next resource is called the L1000 Connectivity Map, or CMAP for short. This is another project that's part of the broader LINKS project. And what the L1000 CMAP project is doing is it's profiling gene expression changes following pharmacologic or genetic perturbation of cell lines. So what I mean by pharmacologic perturbation is treating a culture of cells with a drug. And a genetic perturbation would be modifying the cells by treating them with an agent that's either going to knock down expression of a particular gene or cause increased expression of a particular gene. After these perturbations are applied, then the gene expression of these cells is measured, and then this can be compared to control conditions to determine how perturbing the cells in a particular way causes the expression of the genes to change. Another resource where you can obtain this kind of information is the Gene Expression Omnibus. This is a bit of a different resource in that the Gene Expression Omnibus is just a repository for many, many different groups to contribute data sets that they've generated studying particular conditions, whether they've been interested in studying what happens when you knock down a particular gene, or they're looking at a particular disease state and comparing that to control. In all these different cases, you can obtain a signature of what are the differentially expressed genes when you compare a particular perturbation to control. So this screenshot on the right is showing you where you can go to learn about and get access to the LINKS L1000 data. And on this next slide, I'm showing just a little piece of the data. Here we have a gene expression matrix like we've seen in a few previous examples. The column labels correspond to particular samples where we're either measuring gene expression in a control condition or in a treatment condition, which again would be a drug treatment or a gene knockdown or gene overexpression. And then the row labels are telling us the genes that we measured the expression of. And so the values in the matrix are quantification of the expression level. And so what we can do with just this one set of data that's shown here, where we have a group of control samples and a group of treatment samples, is use a statistical method, for example the characteristic direction, to compute which are the significantly differentially expressed genes between control and treatment. Once we have this signature of the differentially expressed genes, we can combine it with many, many different perturbations. And on this next slide, we see a heat map where we get this broad picture of how many different perturbations are related to each other based on the gene expression changes that occur after applying the perturbation. So all the columns in this heat map correspond to different perturbations, in this case gene knockdowns, and then the rows correspond to the genes that we measured the expression of. And so red regions of this heat map correspond to genes that increase expression after gene knockdown, and blue regions correspond to genes that decrease expression. This next resource is called the Mammalian Phenotype Ontology. It can be accessed through this website that's shown in the screenshot on the right. And this was developed by the Mouse Genome Informatics group at the Jackson Laboratory. And what an ontology is it's a controlled vocabulary. In this case, the vocabulary is composed of mouse phenotype terms, which are broadly one of two things, either a morphological description or a physiological description. A morphological description, for example, might be, a very general one might be abnormal heart development. So what's done with this ontology is that phenotype data for all of these different transgenic mice are curated, and if a particular phenotype was observed for one of these transgenic mice, then that particular genotype would be assigned to that phenotype. So a genotype might be a gene knockout that was performed in a mouse. So what we can look up from this data set is, if we're interested in one particular gene knockout, we can look up all the phenotypes that have been associated with that gene knockout. On this next slide, we see what the data set looks like. This is just a list of associations where we have two columns that are primarily of interest. One column has identifiers for phenotypes, and the second column has identifiers for genotypes. Again, this would be correspond to, for example, a particular gene knockout that was performed in a mouse. So we can parse this data and reorganize it into a matrix like we've been doing with these other resources. And in this next slide, we see the heat map where the rows correspond to genotypes. In this case, it's just showing gene knockouts, and the columns correspond to different phenotypes. And so what we see with these red spots on this heat map is where particular gene knockouts cause particular phenotypes.